Human Genome Project started in 1990 and was finally finished in 2003. But in reality, only 92% of the genome had been sequenced because no technology at the time could accurately sequence the long stretches of highly repetitive DNA in the remaining 8%. Only stretches of about 500 nucleotides could be read at a time. As a result, none of the centromeres or the short arms of 5 of the chromosomes could be sequenced.
I'm limited by the technology of my time. But one day you'll figure this out.
In fact, David Dreamer of UCSC had already started down a path to a solution even earlier, first sketching out the idea for nanopore sequencing in 1989. 25 years later, the first portable nanopore sequencer was released in 2014. In May 2021, less than 2 years ago, sequencing of the human genome was finally completed with the help of long-read shotgun sequencing technology from Pacific Biosciences and Oxford Nanopore. Third-generation technologies such as nanopore sequencing can generate reads spanning hundreds of kilobases in real time and at lower cost than older next-generation technologies such as Illumina paired-end sequencing. So does that mean that nanopore sequencing makes Illumina sequencing obsolete in the same way that Illumina sequencing made microarrays obsolete? Well, no. In fact, the technologies are highly complementary, and both are needed for large-scale sequencing efforts. The problem is that long-read sequencing is extremely error-prone, with an error rate of up to 10%, whereas paired-end sequencing produces short but extremely accurate reads. Long-read sequencing is often used to create a scaffold, and paired-end reads are used to resolve the final sequence. Oxford Nanopore produces a range of devices, from the ultra-portable flongle with a single flow cell to the promethion with up to 48 flow cells. The flongle and minion are particularly well suited for viral surveillance. Nanopore sequencing has played an essential role during the COVID-19 pandemic, with a quarter of all SARS-CoV-2 genomes having been sequenced using nanopore flow cells. Because each read can span the full viral genome, the method is useful for identifying viral haplotypes. With older methods, when substitutions occur in different parts of the viral genome, it's hard to tell if they co-occur in the same sequence or if they're present as different quasi-species. This is particularly important in the case of antiviral resistance. Mike Onamba in the Department of Gastroenterology is using nanopore sequencing to identify deletions in the pre-S region of hepatitis B virus. Deletions in this region significantly increase the risk of hepatocellular carcinoma through ER stress and activation of pro-inflammatory pathways. She extracted DNA from serum from 6 patients at a time and added barcodes to identify each patient in a pooled library. In principle, one of the advantages of nanopore sequencing is that no PCR amplification step is strictly required, but it was needed in this case to extract enough viral DNA from the patient's serum. After DNA repair and prep and adapter ligation steps, the pooled DNA library was injected into the flow cell. Inside the flow cell, protein nanopores are embedded into an electrically charged membrane through which ions pass. The DNA adapter sequence attaches to a tether, and a motor protein ratchets the DNA sequence through the pore. As the DNA moves through the pore, it disrupts the flow of ions. This causes characteristic changes in the current that can be used to identify the sequence of bases. In this case, the machine generated about 500,000 reads per run. Reads were separated by patient using the barcodes, and the adapter sequences were removed. We used an alignment tool called Minimap2 to align the reads to the HPV genome, and a tool called NanoSV to identify deletions. From profiling somatic variants in a tumor to monitoring wastewater to head off the next pandemic, there is an ever-increasing need for sequencing in medicine and public health. Third-generation sequencing technology from Oxford Nanopore and Pacific Biosciences have revealed long-hidden parts of our own genome and brought sequencing capabilities out into the field and up to the front lines, where time and budgets are often limited. Thank you very much.